Okay, so so already 7:08. So I start. Okay. So I hope my slides are visible here, so that I can also have a look. Okay, so slides are not yet visible. Is the slides visible? Hello. Hello.
Nope.
I I can't hear you and hear you.
The screen's blank. Yeah, it's not visible.
Slides. Hello. Am I audible? Hello. Uh, Slides are not visible. Okay, slides are visible, right? Okay, fine. Uh, okay, and can someone just speak so that I can have an idea that I can hear you also? Hello. Can someone just speak or something like just say hello or I can also know. Easy for me. It's not visible now. Okay, fine. I'll just
Hello, sir.
do one thing. Stop sharing. Share. Share. Okay. Okay. I think there is some problem is happening. 1 second. I'll just stop this sharing and do screen share. Oh, I think now. Yeah, now it is visible. Okay. Cool. Yeah. Okay. So good evening, everyone. So I am Shobik. So I am an integrated PhD student at Indian Institute of Science, Bangalore, and I am doing my PhD in molecular biology in Department of Biochemistry under Dr. Sandeep M. Ishwarappa's lab. And my work is mainly like focused on translation read through and translation in overall in the system. So translation, as you know, the protein synthesis and translation also will be a topic that will be covered in the course later. So this is a small like a view of the institute's one of the buildings, and This guy, this here is actually me. So let's go to the yeah. So outline of the class. So uh, the basic need of this, or basic the thing that will be done in this problem solving sessions will be to discuss about any doubt. So if you talk something and you are feeling any doubt, so you can always like clear it here in the same in this class. And if you have any doubt that you want to like you couldn't would have want you don't have to have, you can also mail in this in my mail ID here at the showbiksham at that iic.ac.in, or you can always post in that forum where all that there also I can see and I can answer your questions. Okay. So yeah, the main focus should be clearing any kinds of doubts. If you have anything that you want to clear in this class, or in the you have not understood in that in the videos, that should be come up in the session. And always try to ensure like always make sure that like don't think any doubt is silly or whatever you have in your mind, you can just ask. Okay. Okay. And second thing is that so one thing I will be doing to help you solving the assignment. So I will solve some of this previous year assignment questions. And so most of these questions will be of actual types, because that's how the course actually is designed. And there is nothing much to describe, but if anything I can describe or I can elaborate, which could be or may not be in my field of expertise, I'll try to do. Okay. And this is one thing I also wanted to do. So this is a biology. Okay, so biology is all about, and not only biology, science is all about invention discoveries, right? So whatever week's related content, we can discuss some of the interesting discoveries related to this week's content. So from this, I am planning to do from the next class to make this class more interesting. And if feasible, and if you guys are interested, or if you guys have some knowledge of biology or little knowledge of experiments, then we can discuss some of the experiments which we have has like had won Nobel Prize, or had very long term effect in the evolution of biology itself, the biology subject itself, or evolution of medicines itself. Okay. And yeah, so anything else if you want to add on this, so you can also add. So yeah, before going to the next next slide, I want to just in a short period of time I just like to know that so what background you people are. So how many people have at least had like biology, like okay, first of all, how many people are there if we are in like within 12 standards, or like all the people are here or above college? So can you just okay, people who are in the say below 12 standard, can you please raise your hand if you are like in class 11, 12, something like that? Okay. Please raise your hand if you are in like school level or pre-college, pre-university level. There is no one. Okay. And is there any okay, so so people who are in like pre-university level, sorry, in the university level, like are there in their BScs or masters, can they raise, please raise their hands so that I know that what is the formation of the class is, what is the phenotype of the class basically is. Anyone can raise their hands. Okay, so from college. Okay, so most of the people are from college, right? Okay, then good. Then we can always discuss some experiments, right? Because you people are doing experiments and all. So from next class, I'll try to add some more things. So in this class, this since it was mostly like taxonomy and evolution, there was not much chance to do like much of evolution like much of the experimental things, because I myself is a specialized in molecular biology, so I won't be able to will also follow that. Okay, so that's the good thing. Okay, okay, you can raise down. Okay, you can uh, raise down your head. Okay, okay. So anything else you want me to add here? You can also tell me in the either in the in the message or in like wherever in if you want like if you want to unmute yourself and tell, that will be a very good if you want to do. And so yeah. So first, so main thing in today's class, I'll be mainly discussing some previous year assignment questions, which will help you to solve the assignment questions, and mainly those will be factuals. Okay. Okay. So first question was the smallest unit of taxonomic classification with fundamental similarities is. So what is the smallest unit? So as sir told in the class, there are in taxonomy there are several orders, and so the highest order is eukarya. Then if you consider like Homo sapiens or human, the highest order is eukarya. Highest order is eukarya. Then we have in the human, they belong to animalia, right? There are five domains, and there are plant kingdoms, animal kingdom. So human belongs to animalia, and then next they are human also belongs to chordata, right? Phylum chordata. Then after that, they fall under class mammals. So there are different class, right? If you remember, like fish, amphibians, reptiles, birds, and then mammals. Okay. So human falls under mammals. Then order is primate. Then hominid is a family of human. In genus is Homo, and species sapiens. So the lowest fundamental, the smallest unit of taxonomic classification is species. So for human, the Homo sapiens, correct? Because in Homo also there are other species which are also um which are all extinct, like Homo neanderthal. There are Neanderthals, right? Because they were the like oldest human we have gone which have gone to extinct. So the Neanderthals. And recently, I think you may have, if you have followed the Nobel Prize, you know this year, Papo has got the Nobel Prize for sequencing this Neanderthal genome. And I'm not able to remember now. There is another species of hominids that were also hominids. So these are the hominids. So another species of hominids uh, that was also there. Also, he has invented. So I'm forgetting the name. Anyway. So okay, the answer is species. Okay, now the second thing. So yeah. So the ribosomal RNA present in both prokaryotic and eukaryotic ribosomes. So which of the subunits is present? So let's see here. So I have a diagram here, and so small subunit and a large subunit of both prokaryote as well as eukaryote. So so the prokaryote, the subunits are 30s and 50s, and combining that will make you 70s subunit, and 60s and 40s subunit, which will combine to get a 80s full complete subunit, right? So this 30s subunit has made up of 16s rRNA and ribosomal protein. The 50s subunit is 23s rRNA, 5s rRNA, and the 34 proteins, correct? So, and for eukaryotes, the 40s subunit is made up of 18s rRNA, and the 60s subunit is made up of 20s rRNA, 28s rRNA, sorry, 5.8s rRNA, 5s rRNA, and 50 proteins. So which is the common rRNA? The 5s rRNA, right? 5s is here also, 5s is here. Okay, so 5s uh is the 5s is the correct answer, right? Okay, so next question. Okay. Uh, so association between two organisms function as a single unit and help each other in other for survival, which is that is called. So they are helping each other. So they are both like the species one and species two. Both are positive, correct? So is it symbiosis, asymbiosis, amensalism, or neutralism? So let's see our here. So in competition, what happens? So competition means I am so one species is competing. So same species one sorry, two species is competing with each other, right? So for example, there is a deer, and some tiger is also going for it, and some the jackal is also going for some deer, or some hyena is going for there, right? So that means in this situation, neither the tiger is benefited nor the jackal is benefited. Both are negatively benefited. So this is a competition. Okay. Then mutualism. So mutualism means both species are having positive effect. For example, there is a flower. There is a flower. Oh, interesting. So I have a flower, and let me try to clean this. So I have a flower. So in that flower, there is some insect is coming. So what that insect is doing? So this insect is getting nectar, and this flower is having the pollination, correct? So that insect is having nectar and pollen. So this is both the species are positively affected. So that is a case of mutualism, correct? So okay. Then next is that, what is that? Antagonistic, or like what is antagonistic now? Say there is deer and tiger is predating on that. Okay. Or there is a human, there is human, and there is mosquito. Correct. So in this case, tiger is negatively affecting the deer. So tiger is having a positive effect, but deer is having a negative effect. Again, in mosquito, they are getting blood from human, but human they are negatively affected because they are getting diseases. So both of these are, this is predation, and this is parasitism. Another example of parasitism is parasitic plants, right? Parasitic plants. So the big plants that is getting nothing, that is negative. You are not getting nothing, that is negatively affected, because it's whatever nutrients that big plant is getting, it has been taken by this parasitic plant, and so so predation, parasitism, all of these are antagonistic. Okay. Then amensalism, where amensalism. So amensalism is there that one species, correct? Sorry. So species one is have be species one is negatively affecting, and species two doesn't have any effect. So there is a very good example is like insects. When an elephant or anything is walking, it is walking and crushing the insects. So insects are negatively affected, but this elephants doesn't matter. Even some insect dies. Okay. Then is very good example is commensalism. Okay. Commensalism is that the species one is having positive effect. Species two doesn't have any effect. Correct. One of the example here is given as shark. So remora, there is a small fish remora. So when the big shark is going through the sea, okay, the big shark is going to the small fish remora. They generally get onto this shark, and shark from outside there is no positive. Shark doesn't from outside don't have any positive effects for this. So remora, so if this is the case of common Sennheiser, because this species remora is getting positively affected, but this other species shark doesn't have any effect. This large shark doesn't have any effect. Okay. Okay. And last is neutralism, where like there are two species there, but doesn't have any. They don't have, basically they don't have any interaction. So this interaction itself is not, so species one and species two doesn't even interact. So it's like it's and this kind of for neutralism, there's a very difficult to get a proper example of neutralism, because uh in an environment every species somewhat interacts with other. So it's very difficult to have something that is to have a neutral interaction in the nature. Okay. So there's like two species without niche overlapping in a sharing habitat. So you can say like say one human is there at some place, and say some birds, there is a tree and some bird is here. Okay. So that bird is doesn't have any, like theoretically doesn't have any interaction, because human doesn't give any food to the bird, and bird also doesn't go, and so it's like a very crude example of neutralism, because again it's very difficult to get a example of neutralism in nature, because two species when they share niche they will have some effect. Okay. Okay. And so what is there? Symbiosis. Okay, symbiosis is something that, so symbiosis will come here. Mutualism. So mutualism is, there is another term of mutualism. Mutualism is also known as symbiosis. Okay. So and neutralism is another term of asymbiosis. So those species unit help each other for survival is of course symbiosis. One of the very good example of symbiosis is, I hope many of you will know, like interest. Okay. So let's go to the next slide. You have something? Okay, I had put some videos. I don't know why it is not coming. Anyway, so protein-rich layer in the cell body of eukaryote that gives flexibility. The organism is, it is pellicle. So I had put some video. Let me see if I can. Okay, if it is. Oh yeah, it's not playing. So as you can see, there is a Euglena. I got it from YouTube. And so this is the Euglena. This you can see this flagella, and this is getting flexibility and contractile. So this is this part here. Here this part. I can't. Yeah. This is the Euglena. Here this part, the body wall. There are pellicles, correct? So this helps in this organism to move, correct? So the correct answer is pellicle. Okay. Yeah. So next is like the organism lives in extremely salty areas, that is either halobacterium, thermoplasma, pyrococcus, and sulfococcus. Now salts and halo. Always remember that salts means halo. So anything related to halo is will be go to salts. So of course the correct answer will be halophile. Thermo, as you can say, like heat. So thermoplasma means, that means it stays in very heated environment like hot springs or mud lavas, something like that. Pyrococcus, pyrococcus, pyrococcus. This is something that is not very common. So Pyrococcus, I think, also an extremophile. It stays in not able to remember. Remember, so Pyrococcus is also a thermophile. It should be here somewhere. Oh yeah, it is Pyrococcus, correct? Thermophile. So again, thermo is with thermo, but this is a little difficult to guess because you don't know. Sulfococcus, again, you should know there will be like gaseous high sulfur concentration, and there should be same as sulfuric acid. So there should be acidophile. See here, Sulfococcus, Sulfobacillus, all are here. Okay. So there are different types of extremophiles as I have mentioned here. So one first type is thermophiles. There are several types. Thermophiles mean they are really very high temperature. Okay. Then psychrophiles means they stay at very low temperature. So I was just I was reading this thing and I found that there are two types of there are two different things. So there is one term known as psychrophile and second term there is psychrotolerant. Sorry. Psychrotolerance. Cyclotolerance, correct? So, and there is one more term was there, psychrotropic. Okay. So there are three kinds of. So psychotropic means that they general. So this psychophiles and psychotrophs, there are two differences. So these actually generally generally these people grow better greater than 20 degrees Celsius, but can grow slowly at less than 20 degrees Celsius also. Can grow slowly at less than 20 degrees. These people have optimum growth, optimum growth at less than 20 degrees Celsius. So you can see the difference. These people generally grow fast at 20 degrees Celsius, but they can also grow slowly at lower temperature. But these people have optimal growth at 20 degrees Celsius. And psychotolerant, these people doesn't grow at all at maybe less than 30 degrees Celsius, but they can withstand that high stress with some mechanisms are there. With that, they can at least uh, tolerate the cold temperature. Okay. So next is metaphiles. So they generally like stay in highly metallic environment, kind of close to like this alkalophile, acidophile. So acidophiles are which stay in the highly acidic environment, like some of this lake which are very acidic and some lake which are very alkaline. So maybe are found here. So there are some examples, and you can also always go and have a look on them. And yeah, and then you can see your halobacterium should be somewhere here. Halo, yeah, see, halomon, pseudo, halo. So most of this salts would be make the yeah make the area like many of the halophiles will be also there. So many of the salts that make the water very alkaline, that will have those halo will be there. But mainly halo is like salty. Radiophiles which can have like radiation resistant bacteria like this. And then you have halophiles which stay in the salty areas like halobacterium, I think haloborum, halococcus, some lots of halo is there. So the correct answer is halobacterium. Okay. So yeah, next question is left. So molluscus uses their feeding organ called radula. As you I can show, I've shown you. This is the kind of a molar or snail. This is the radula here, and radula is like enlarged, and you can see there are like teeth-like projections here near the radula. So what is radula made up of? Again, this is like very factual question if you know. So it is made up of chitin. Okay, and there is a structure of chitin. So chitin is a uh, long chain polymer made up of disaccharides like uh, polysaccharide. Sorry, made up of this polysaccharides of N-acetyl. So this sugar next class you will be seeing N-acetyl glucosamine. Okay. So at N position, there is a glucose and amine. This is the amine group. Okay. He is the amine group. So an acetyl glucosamine. And there is the acetyl bond. Okay, so show you. So this is the amine group and this is the acetyl bond. So this is the that's why it says N acetyl glucosamine. Okay. Okay. Compound eyes are present in mosquito, octopus, frog, and humans. We all know this is mosquito. So this is one of this like basically arthropoda have this compound eyes. Okay. And there are some very interesting papers actually how many interesting studies that how actually so our eyes we are simple eyes, right? So in compound eyes they will have multiple lenses and lenses, right? So how this multiple lenses actually focuses light, there are good studies beyond that because you have to focus the light in from the multiple because for our case we have only one lens here, correct? This is our lens, this is our cornea, and if this is our eye, and here our optic nerves is there. So if this is our cornea, the light is passing through the cornea, then to the lens it is falling and it's getting like focused at some depending on the how from where the light is coming, generally it focuses on some point. Okay. If the light is coming like this or this, whatever, it can focus at some point. Okay? And this focal point would be some in the retina. Okay. Next. So yeah. So the coiled tubular duct organs which filter and remove waste from earthworm is again this is a very factual question that is known as nephridia. Okay. Protonephridia are present in some like platyhelminthes, aschelminthes in those cases. But earthworm, it's the annelids. It's present, so it have this nephridia. Where is nephridia here? Yeah, here is the nephridium. Okay. This is the part you can see nephridia. Okay. So again, yeah. Okay. The egg-laying mammal with a beak like a bird is which is this example. So okay, let's see. So duck has a beak like a bird and egg laying, but it is a bird. Kangaroo, kangaroo doesn't have a beak like a bird, but it's a mammal. Duck is not a mammal; it's a bird. And kangaroo is, as you know, it's a marsupial, right? So it carries its baby till its maturation. King cobra, right? King cobra is a snake, as you all know. So that remains echidna. Okay. So the correct answer is echidna. So this is one of the beautiful thing there is the picture of an echidna. You know, And yeah, so this they lay eggs, generally echidna lay eggs, and their beak is like a bird like beak here. So these people generally they are ant eaters, so they don't have any like birds. Like birds, they don't have any teeth, okay? No teeth. And they use their they have a long sticky tongue, kind of like a frog, like the long sticky tongue to like if this is a uh, ant nest, which will use its long tongue to go inside. And since it's sticky, the ants will and some other insects will come and then it will go and eat it. This is known as echidna. So they legs and they have a beak like a bird. Okay. Next, the characteristic feature of heart and pattern present in mammals. So yeah, incomplete or three-chambered and double circulation. This is not correct. So who have incomplete and three-chambered double circulation? Someone tell me. This is something like very. I think in the course it has been told lots of times. Someone tells me who have this incomplete three-chambered and double circulation. Can't hear you. Hello, I can't hear you. Can you speak louder? All right, it's my issue. This is my issue. Okay, so incomplete chambered heart and double circulation, complete two chambered and single circulation, then incomplete fourth chambered and double circulation, and complete fourth chambered and double circulation. So which are those? So this, as we all know, is PBN. Complete two chambered. There is complete chamber single circulation is not there. Incomplete fourth circulation and double circulation is yes, guys. And complete fourth chamber and double circulation is mammal. So the complete fourth chambered means like the heart. So in this case, there will be a complete four chambered heart. Right, like this. So this is the heart wall, and there are septums here, so that heart abundant blood cannot pump. Correct. And here is the so so here so one side is so it is the left side of the heart where it is like oxygenated. Left side of the bird where it is like oxygenated blood will come, and there's the so this blood will come from here. This is uh, the vein that comes from lungs, pulmonary vein, correct? Pulmonary vein. Pulmonary vein will come with oxygenated blood, come here and go to the vena cava, right? And then this is the here from here the carbox carbon dioxide concentrating containing blood will come this part. Oh, like it's not sorry this part. Sorry for the disturbance. Yeah. So where were we? Yeah. Okay. So in call for chamber. So yeah. So I was drawing a part of the picture and here and some system here. Yeah. So the carboxylated blood will come like this, go and go out to the into the uh, lung. Okay. So complete fourth chamber and double circulation. Double circulation means that we have both veins and arteries. So we have double circulation, our blood zone means. So complete fourth chamber and double circulation. Okay. Type of scales present on the Bombay duckfish. Now it's clearly written it's a fish, and Bombay duckfish is, as we know, it's a bony fish. Correct? It's a bony fish compared. What are the cartilaginous? There another thing. What is the cartilaginous? Cartilaginous. Yeah, I'm sorry if the spelling is wrong. Okay. The cartilaginous, which are the example is shark. Correct? Cartilaginous fish. So which kind of scales? Now, placoid scales, cycloid scales, placoid or tenoid, cycloid or tenoid, or only placoid scales. So what cut is kind of different kind of scales? I have a picture here. So bass or those fish, they have this kind of cells, which is tenoid scales, as you can see. I don't know why this is called tenoid, placoid, or cycloid. I have no idea. But anyway, since it's a bony fish, so bass is also a bony fish, and salmon is also a bony fish, correct? And this ganoid is not. Okay, ganoids are present in some of these bony fishes only. There are in some few bony fishes present, and mainly this tenoid scales are present in this parts and this part, and placoid cycloid scales are mainly present, I think, in the body. And this is the shark, which are placoid scale. So we have a bony scale. So bony scales have both tenoid and cycloid. What will be our this one? Cycloid and tenoid. Okay. Because they don't have placoid, because there's only placoid also. Okay, so now we'll come for some example assignments questions from week 2, and that is like the evolutionary biology part. And let's see what is here in the week 2. Okay, so week 2 was again more like a theory based. Like I don't have much to explain here. So okay, so what theory is stating that God is the creator of the earth and organisms? So theory of eternity of life. So what they told the eternity of life, that life is like indestructible, correct? Life is indestructible. It was already there, and it was, so this is not God, the creator of that. It was like life is indestructible. It was always there. Theory of catastrophism. So at some point of time, so like somehow the God has destroyed all the animals, and again renewed all these things. So this is the correct answer, I know. The theory of catastrophe. Cosmozoic is no like coming from outer space. Mm, outer space. And modern theory is the chemical evolution theory, correct? So this is what we have know, that this is the most important, like the most important and mostly well explained theory, right? Modern theory. Okay, the Primitive of one cool down the below 1000 degrees Celsius was led to formation of biosphere. Not possible because at 1000 degrees Celsius, no biological organism can survive. Ecosphere again, it is not possible. Magnetosphere, it was already there because magnetosphere is at the like near to the core of the earth, not the core, just above the core, magnetosphere, which is also not possible because this temperature is quite high itself. And so one thing only can be done is hydrosphere. It's like the water like region. So hydrosphere. And okay. So the part of the with the large quantity of iron and nickel. Okay. So which part? Okay, this you should tell, like, sir. I think there was a image like this here in the image, and so there was like inner core, like this is outer core. So this is the what is known as perisphere, and then there is. Oh, I think then there was the like, this pyrosphere mantle, and then above was the lithosphere. So iron and nickel. So which one was from what the iron and nickel? I want to know from you guys which one. For some like this is a very common question. And this will be in the outer core, right? Or inner core. This is the outer core. Unit of Benz. Okay. Next experiment is to disapprove the theory of spontaneous generation. Louis Pasteur with the experiment involving S-shaped tube had given the following analysis. Appearance of microorganisms. So S-shaped tube, as you remember, so they wanted to prove that spontaneous generation of life. They're trying to disprove that. What he had a flask with some broth, and he had made like a tube kind of like this. So he boiled it, and then to make all the air out, and then let it cool down, correct? So at this position, all the condensed water will come. So the and air can also pass through it. Only they could find that there is organism in the condensed water, but not in the this part. So they could see that there are some bacteria here at the condensed water, but nothing was growing in the. So yeah. So what are the so appearance and disappearance of the. So appearance and disappearance of the appearance and of the microorganism at the neck of the condensed water. Disappearance of microorganisms in the neck of the tube of the condensed water. Appearance of inside the flask with condensed water inside the tube. Correct. So appearing in the this one, correct. Appearance of microorganism in the neck in the tube with the condensed water. So this part. Miller and Wood experiment. So we know this is the correct answer. It's again something very mugging up things. So Miller and Wood, they had a experiment in a flask and they put some gases here, like this gases, ammonia, uh, methane, ammonia, and hydrogen that was present in the that kind of environment will be there, and there would be some lightnings. There will be some lightning spark like that will be do like electric sparks that they use that like proxy for lightning, and they would see that compounds are forming here. Okay, so this was the ratio that we have. According to the chemical theory, the energy of formation of inorganic molecules are provided by the energy at that time for organic molecules are provided. Of course, there that time there was no ocean, no there was lightning. Yes, there was no plant, no electricity. So, lightning. Okay, so the which era doesn't have any fossils? So this I have given a how the earth is divided. So this part is the younger and this part is the older. So as we know that like all the fossils started coming from this particular area. This is like uh, what the Proterozoic era, but there are very few fossil at this particular area. Most fossils start at doing from this Cambrian era, this part, Cambrian era. So before that, like 4.4 billion trillion galleon years. So there was no fossil. So this Archean part, there was no fossil, but there are some like examples of like archaeas. So as you know, there are like so for example, there's archaea. So archaea, these are so most of these extremophile bacteria are extremophile. Those have been generated in this Archean era, but there is no uh, fossil evidence. So actually, the Cenozoic is a very the recent era, correct? Cenozoic, and Mesozoic is the Jurassic area, the when the dinosaurs came. Paleozoic again this that Cambrian area and all that time, which have a lot like which start getting the fossils. Before that, we don't get the fossils because for getting the fossils, mostly you need the sedimentary rock, right? Sedimentary rocks and sedimentary rocks were not available in Archean era. Okay. Okay. Who disapproved the theory of inheritance of acquired characters? Of course, August Weismann. So what he did, he cut the tails of mice for so he to what Lamarck told. Lamarck told that uh, inheritance of acquired characters, right? Now, so that time there was no concept of two types of cells, right? So which are there was no concept of two types of cells that were as somatic cells, somatic cells, and germ cells. So, so Lamarck thought that whatever uh, character that has been acquired by the somatic cells or germ cells, that would be inherited forward. So somatic cells, as we know that these are cells that is the body cell, they don't get inherited. Okay. So but the germ cells, they inherited to the next generation. Correct. So in case of tail cutting experiments, all this thing was done in the somatic cells. Right? Tail is a somatic cell. But think that Wisman, if he knew and if he had done the same experiment in some of the gonoidal cells in the gonads, he may have actually like subside like may have actually proved that okay, Lamarck was not fully wrong, but he didn't know that the character should be acquired in the germ cells, and this acquisition of this characterization is done by spontaneous mutation, right? Spontaneous mutation. Which who proved? Who got replaced, correct? Spontaneous mutation. Uh, so what is the link between the cartilaginous fish and the um, bony fish? Silicon or chimera? Phenodon neoplane. Okay. I thought to put one. I forgot to put. I thought to put one images of this each of this what, like, the or each of this because all of these are see all of these are like missing links of some. So silicon is a missing link for fish and amphibians. Then phenodon. Phenodon. Phenodon is a connecting link between amphibians and reptiles. Neopalina. Neopalina is a connecting link between a non-vertebrate like annelid and mollusks and chimera. So chimera is actually the connecting link between the cartilaginous or like connecting between this cartilaginous. So correct answer is chimera. So what are those missing links? Missing links are like so as we know that the evolution says that the development of evolution says that development of species are are in order of lower to higher animal, right? Lower to higher. Lower to higher, maybe lower to higher complexity, correct? So there's missing links. So now this missing links, they are the things which have features common to both. For example, platypus is a missing link, correct? First is a missing link between bird and mammal, correct? The missing link between bird and mammal. So like that, different missing links are there. Okay, Which of the following is false regarding the mutation in the offspring of Fukudibri's experience? Which one is false? Okay. In the Oinothera lamarckiana. Okay. Constant and plants are stable. Absolutely. There are mutations which makes the population is like regressive with lost traits. Of course, it can be regressive with lost traits. The appearance of mutation. The mutations can be if some trait is lost, then the mutation is regressive. If the mutation is a newer trait is seen, that means the mutation is progressive. That it has like if you see a phenotype, that means the mutation have some advantage or can survive. Degressive with weak and low survival. So if you see that for some mutation, some uh like some for some mutations, if the animal or if the plant is having survival disadvantage, it cannot survive. That means this is a digressive mutation. Okay. So yeah, these are different kinds of mutation. And yeah. So with this for today's session, this was what I wanted to tell. So now I'd like to ask like if any of you have any doubts and if you have any questions to which I can answer, I'll try to answer. Even if you cannot answer, I can try to. But lot it was sleeps. Oh, sorry. Sorry, it's my bad. Uh, sorry. Yeah, platypus is not between birds and mammals. Sorry. It's a I got confused. Sorry. It's my word. It's between reptiles and mammals. Yeah. Yeah, birds and this is it's between reptiles and because yeah, if it lay egg, it's stay in water. It can yeah, it stays in water and lay eggs. And we are the again here are part like big also. So between birds and mammals. So what is the so I don't you remember now. So what is the between what is the connecting link between birds and mammals? I don't know. Check. Okay, forgot. Link link between birds and mammals. What is the birds and mammals? Okay, Semoria is the connecting in between aves and mammals. How does Semoria looks? I don't know. Oh, Semoria. Extinct genus of terrestrial tetrapods. So this is extinct. Okay. So extinct genus similar to extinct, although they were amphibians. Yes, but it's not written here that. Okay, I'm not sure if it's like a if it is like it's a connecting link between birds and mammals. I'm not sure. This I'll check once. But what is the connecting between birds and mammals? Thanks for pointing out. I also confused got platypus because platypus has like they lay egg and they have like a duck like a bill. So this is how platypus looks like. This is how platypus looks like. Platypus. Yeah, they have like a see they have like a very uh beautiful beak like ducks, right? See. Anyway, so yeah, for today's class, this is how like this is what I had to say. The next week, what is the uh, so next week? I think the topic is, if I'm not wrong, is cell biology. So next week, I think is the topic is cell biology, or so yeah, cell biology is the topic. So yeah. So in this part, cell biology part, I hope I can explain some of these experiments and some interesting discoveries in the cells that have actually made the cell biology so much interesting and so much diverse, and how our cells work. So this kind of like I have some knowledge because again I have done my bachelor's in life science with specialization in cell biology and molecular biology. So and my research work is still now also in cell biology and molecular biology. So let's see if I can frame some like I can show you some experiments which previously they have done. I mean, not demonstration, but slides and slides. Which previously people have done and they have shown that okay, with this experiment that this has been proved. Let's see if I can show you that. Okay. And if there are some Nobel Prize winning experiments also, if I can show. Sir, what is the consequences of? See, as per I know, last year I have taken one like TA of the one course. You cannot submit the if you cannot submit the assignment, I like you will not get the marks of that particular like uh number of that particular assignment. I don't know if there is best of three or best of five. I'm not sure if in this course it is there, but how does NPTEL online work? Let's see. I don't know. I'm not sure how to like what will be the this kind of questions you have to ask to this NPTEL coordinator. But the most main thing is that if you cannot submit on time, the portal will close and you cannot do late submission. There is no option of late submission because the portal will be automatically closed off for submission. Okay. And so if you can't submit, you will lose the marks. That's all I think. Yeah. Multiple flows running together. So that's you have to manage, right? Okay, anything else you guys want to ask or I have to say? So just okay, we have some time. So just tell me what do you expect from this particular TS session because I should know what you people should why people are you are expecting. Solving assignment, yes, that's like one part, but in 2 hours we can do like lots of things, right? We can discuss lots of things. So what you want me to like do? Hello. So yeah, I want that thing is what you guys want me to do. Like I want some inputs from you also. That if you say something like, okay, I want you to describe this. If I can do, or like if I have the knowledge to do, I'll try my best to do. But you people should tell me that why. Because from the NPTEL, they have told us to um they have just told us to do a like do a assignment solving session. But in this assignment there is no solving, right? So they're very much factual. So I just want to know that what you people want to have in these sessions. This is something that I want to know. Anyway, you can think and let me know if you have any ideas. You can also mail me. I have given you my mail ID, right? So I'll also type it here. So the typing, I'll also type it here once so that you people can also mail me my mail ID. So you can always mail me in this ID and let me know if you want something. Okay. Okay. Then see you guys. And then in next week, next which date we are going next for next Tuesday will be on. So 7th, 14th. So next 14th. Let's see what we can do. Okay. Thank you. Thank you for joining.